Okay. Rahubat. Rahubat. Okay. So we missed a few people, but hopefully people will log back on. Um, while I think the link. Yeah. Okay. So now I see the chats. I didn't see it before. So please share real quick for everyone that wants to join and catch this lecture. Uh, this is the official Actual Facts podcast and live stream where we will address specific topics as it relates to the information that our father, our spiritual leader and teacher and guide, known to us affectionately as His Excellency Dr. Malachi Z.K. York, our Heavenly Father, the Father in flesh, God in flesh, as we know Him to be, for those that are aware of this supreme being and divine being. There's a series of publications that has been written by Dr. York, right, since 1967. And we have an obligation and a duty to teach this information to the world, to teach this information to the people. And there's Africans, Blacks, Nuwapians, people of the diaspora worldwide that need liberation mentally and spiritually. And so our responsibility is to make sure that if they can't get the information that is taught to them and it's available to them through the specific channels and platforms that now the internet has brought the world with social media and the various apps and live streaming applications that are available to the world. So for those that I know what happened. I sent out a link earlier, and um, I had to end it because the chats weren't on. So now there are people that probably are clicking the link and don't realize that we're going live. So hopefully they can catch it at the next go around. But that won't happen again. I just didn't know that the chats weren't on. But anyway, this will be downloaded and uploaded on a podcast feed for people to catch it, for those that didn't catch it. And then with the editing, um, we will have what you call the pictures and diagrams and things available for this information. Now, this Origins of Man um, lecture is kind of it originated from the many topics that people uh, inquire about on the various apps. You see TikTok, you see obviously YouTube, you see Clubhouse, and on these apps, right? What happens is people begin to discuss certain topics off the cuff. They'll begin to try to explain and go into detail about scientific information that they may not have studied. They may haven't did they have not done their homework as it relates to these topics. And it's very crucial that when you have these platforms and you receive people's ears and their attention, their eyes to focus on, okay, what is this individual saying? That you actually have the information that will be available for them. And so this is the platform that you can get the actual facts. You can get unadulterated, pure information that leaves no room for doubt and no room for what you call mishaps, right? Or misinformation, or lies, or conjecture, or beliefs, or philosophies. A lot of times, when people are relaying information nowadays, they come from an emotional aspect, they come from an emotional field, or they come from a space where because it sounds good that it can be relayed to people, and those people will digest it and take it as facts, when really what it does is confuses individuals, and now people are under the impression that anything they feel is about a specific topic, if even if it sounds right or in their mind it sounds good, that they can regurgitate it. And this is how belief systems are really perpetrated throughout the world. Belief systems and doctrines that hold no relevancy, really holds it doesn't hold any factual basis. And because the day and age of information is it's very important that, right, people that, especially this generation, because their attention span is very short, right, it's vital that any information that you take in or information that you take in should be coded in facts and evidence and confirmables. You can confirm it through examples, through charts, through data, through any type of scientific experiment that will basically confirm the information to you, so that you will know it's real. And this is the purpose of this podcast, this actual fact live, um to eliminate the foolery that is the ignorance. It's a sea of ignorance out there, and our people are basically waddling in it, and they have no life jacket, right? So if you're looking for a life jacket, the actual facts, we have a master amongst us. We have a being who has incarnated in flesh for the Nagaru race, the Nagaru race, the descendants of our ancestors, the African people, the Negroid people, the supreme race on this planet, right? And with that being said, we have a specific doctrine, and this doctrine has always been with us in our genetics, right? However, because we have mixed in with the Mukasu, the Caucasoid, and other races, and their cultures and their genes and their way of life, their tones, their foods, their rituals, their customs, right? What it has done, it is clogged, or let's say covered our original genes and source that by which we are connected and linked to our ancestors through our divine blood. And because we've converted to these specific doctrines and worshiping and acknowledging these other races, and acknowledging these individuals and cultures who are foreigners to us, or has always been foreigners to us in our land, has always been foreigners to us by way of our culture. What happens is we take our spiritual powers and we link them to theirs, and we empower them by acknowledging their spirit forces. Whether these spirit forces abuse us, whether they oppress us, whether they destroy us, all of the atrocities that we've experienced from foreign races that entering into our lands, particularly Africa, right? We've experienced a massive amount of genocide, ethnocide, all types of violations, natural law violations, international law violations, right? Economical law violations. Uh, the list goes on and on when you get into the legality part, but the spiritual violations are insurmountable insofar as a 6,000 year spell has been placed on our people in such a way that it's very difficult for us to grasp anything outside of what they taught. And because of that, we always hinge our ideology or our doctrines or our belief systems on what they've laid out in their dictionaries, what they've laid out in their so-called authorities, in their books, in their reference books, in their law books, in their whatever theology books, their biblical texts, their Quranic texts, their King James version of the Bible, New World Translation Bibles, the Catholic Bible, these particular records of their history. We have picked them up and made them a part of us, ignoring our spiritual science, ignoring what our ancestors have given us. And so we'll do our best within these next lecture series to address these particular issues and cure our people with the facts, so they'll know who they are, where they come from. So please, I'm asking those that are listening, can you just quickly share to platforms, and then you're more than welcome to engage in the chat. Um, this is the beginning of maybe a 3-part, 4-part lecture series. I hope you can hear me well. I hope I'm not speaking too loud. If you can engage a little bit in the chat, let me know everything is fine before we proceed. Right now, we're broadcasting live from. Oh, let me do this. We're broadcasting live from Ghana. I have to make sure that my internet is stable. So just bear with me. Just take a second to make sure that the internet. Um, so we have to buy our we have to buy data. It's not internet service where. Oh, okay, let me go back. It's not an internet service where it's just always on and you pay it monthly. No, it doesn't work. It's not with this one. So here I have to make sure that as we stream, uh I think, yeah, this is it. Yeah, this Wi-Fi number. So please share. Wait for a few people to come in. We'll get quite a few views. Now, making sure we have internet so I don't in midstream get just thrown off. That would be astronomical error. Okay. So I think we have enough data. So yeah, so this channel is specifically dedicated to the actual facts, patrak at hakak al hakat, which are the actual facts series, a series of publications that our master teacher, our father, our beloved deity in flesh, who has incarnated for us to teach us and guide us and break the 6,000 year spell. And you see, that's his. I have a statue of him in my home, and right, this is like a chief's palace because in Africa we are royalty. So in our palace we have our deities that we respect and that we love and that we honor and that we cherish. And because we've dedicated our lives to uplifting the Nuwapian people worldwide, and this is for this uh, podcast and channel is for newcomers as well, people who are seeking information, as well as individuals who've been students of the Father since 1967, the first half hour of the hour by which he came to raise us and break the spell. And so the origins of man, this lecture is quite detailed, and it's a lot of science involved in this lecture. And so when we talk about the origins of man, I'm going to first go through the African race specifically, African people, Nagarta Nadu on the continent, the various migrations by which we migrated and populated the planet. We're also going to go through a genealogical study to show how scientists have, or say geneticists, have tracked the haplogroups. Haplogroups are specific types of genes that are in various populations globally, as well as in the DNA that ties you into a specific matrilineal line, DNA line, right, through the mitochondria. We're not going to address the Y mitochondria. We're going to address the matrilineal, right, descendancy, which is the mitochondria DNA that is in the cell that scientists or geneticists use to mark, or uh, let's say I don't want to say mark. I want to say record, right? Record your lineage back from mother to mother or from mother to child back to a common ancestor, right? The common ancestor is the most common group of ancestral individuals, flesh and blood beings that you can be tied to genetically as a race. And this group or most recent ancestor, there's a phylogenetic tree that is that geneticists create that allows this particular breakdown and brings a certain group of people through their haplogroup to line up with it, with this particular most common ancestor. It could be a group of most common ancestors, it could be a tribe, it can be a population in a specific area, but the findings are happens over a period of years, and the DNA tracking through certain sequencing of genomes, and it's a method by which geneticists do this, and we'll go into that, what method is used, what method is typically uh, subscribed as the best, as opposed to old methods that are outdated, and the newer methods that are in use. Also, we'll address the phylogenetic tree that creates branches, that identifies what group of people stem from the root seed, which is the Sans people, the Khoisan people, they're called. They have many names, right? In particular areas, the most concentrated frequency is in South Africa, Botswana, Zimbabwe. We're going to address that. We're going to also address, in the actual facts, how Dr. Malachi Z. York, in his doctrine, right, that he placed on the planet, put on the planet to see his children's minds so they can get a certain amount of intelligence and know who and what, where they come from. In these particular series of books, he has went through uh, verse after verse to speak to us about this phylogenetic tree, the markers in the genes of the Sans people, and how they encompass all of the genes of other races. And scientists now, to this day, are confirming this through this out of Africa theory that has been confirmed, through specific migrations that is being used, the migratory maps that have been, let's say, detailed through computer analysis. They've done that. And also the crossing into Eurasia, the migrations that populated North and South America. We're going to do that. We're not going to address that one in that particular migration, North and South America, as today, because it's pretty long, and that's going to be in part two, where we address what or who the indigenous people came from of North and South America, what seeds they mix with, records, archaeological records, anthropological records, paleography. Will have all of those, and genetic geneticist data, as well as universities and other institutions that are responsible for compiling this data and presenting it to the world as actual facts. So we'll make sure we'll cover as much as that as we can, because it's a lot of science, and we want to make sure that it's not too much in this specific, because I know you're all intelligent, right? Those that are tuning in, and so that's what we're going to address. Also, we're going to address the sources and the references by which you can find yourself. You can look up yourself, look up the authorities. Another thing we're going to address: the chromosome, basic genetic and chromosomal information. We're going to address autosomes and allosomes, which are the sex genes that's passed on, the 22 genes that are your allosome genes, autosome genes, and your one allosome gene. So you have autosome and allosome. Your autosome are your 22 physical characteristics, and then we all have one sex gene. That one sex gene is your Y chromosome or X chromosome that you receive from the father, right, in the mother's DNA. And so we'll address that, and the list of chromosomes, the genes in each chromosome. We'll go through that, and why that's important, because the reason why that's important is because the genes is where the fragmented and the mutations in the genes are records of where you come from. That's how geneticists find out through gene sequencing, right? The common fragment that is available amongst the population, right? That's passed on from mother. It shows a commonality, right, through inference, actually, sometimes, right, through inference, through the descendancy, and how all of those populations, all I'm talking about, even when you go into Europe, right, into Europe, into the Indian, the Hindu Indian area, when you go into the Oriental Asian area, they have a percentage of the L haplogroup, which is the Sans people haplogroup, and the most concentrated haplogroup, which is L, is still in Botswana, and that branches off into another haplogroup, right? So it's two root seeds that creates the 9 species that Dr. Malachi York speaks about in Supreme Beings. So we'll address that as well. So we'll address the haplogroups, and you're going to learn Rahobat. Please share Rahobat. I'm looking at the chat. Thanks for tuning in, and please share to everyone, for everyone to tune in. This is going to be downloaded and edited as well, and when we edit it, it's going to have the pictures, the maps, the dates, all of the details in there. So let's get into and if you have any questions, just put it in. This is our first lecture series for the podcast, so we're going to cover the origins of man. And the reason why this is necessary, obviously, we want to start at the very beginning. Also, we're going to address the extraterrestrial intervention, or the biblical story, all right, that people most the religious world, most of the general population has accepted, that the creation of the planet, of all beings, man, started in Genesis, and this is the record of 6,000-year-old record, and this is the descendancy of all human beings on the planet. Whether or not it makes sense, the illogical reasoning that's applied to it, and in all fields of science, it just doesn't make sense at all. When you look at the genetic diversity of human beings, doesn't make sense. When you look at the blood types or the blood groups of the various human beings on the planet, saying that we come from one person, one male, doesn't make sense. As also the questions of the other beings who were, according to Genesis, Cain's found a wife in a specific land. You had other races in the Bible that have to be acknowledged when you talk about angels or extraterrestrials, or who these specific beings are, like the Anakim, the Pleiadians. You'll see that in Job. You'll see Anakim there. You see the Gibor there. You see many types of beings, right? Haplogroup X. I see you, second. I'll address that too. Haplogroup X and where that group comes from. That's interesting. There's a question in the chat about the mitochondrial DNA coming back to haplogroup X. Haplogroup X. I'll get into that because I have some charts here, right? That goes into the descendancy of where each haplogroup descended from. But all of them come from the sands. And that'll be in part 2 because it's much detailed. Now, when they do genealogical studies, right? They analyze ancient DNA, which is fossils, right? Particularly fossils, um, older fossils, as well as uh, maybe DNA found from, let's say, clothing, which is rare that you find anything clothing, maybe materials or tools or anything. It's rare that DNA really lasts that long outside of the actual fossil itself, because it has a decaying period where it becomes undetectable. But typically with fossils, they go into the bone. If they have bone, obviously in the bone, there's the bone marrow where blood, the DNA is found, and then they can do an analysis on that. With that analysis, typically from the population of where the fossils are found, we'll go into where these fossils are found in Africa, 7 million years old, in Ethiopia, South Africa, Botswana, Tuareg, Chad. You had some in Kenya, Uganda. The father puts a list. There's a list of these fossils, and so they'll take the fossils, take the DNA, and analyze the DNA, and then do charting from the populations and how the amount of trace DNA from this particular haplogroup populated other areas around the globe. Then they will go to these particular areas, get DNA samples, and then do a genome sequence that would determine how much of the fossil DNA is in the population, and compare the two, and then create this huge tree that makes a determination on what groups branched off from there. So it's really intricate, And it's not something that you could just say, oh, I understand it. It's, you have to go through it. And that's what we're doing here. So you have, these are the particular studies when it comes to genealogical studies. You have anthropology, which is the physical. Then you have paleontology, molecular anthropology, and paleogenomics, genomics, right? Then you have, and not then, right? Paleo genomics or genomics, depending on how you say it. It's a discipline that studies, combines DNA sequencing, which we just was talking about, the DNA sequencing techniques with the analysis of ancient biological remains, right? So that's paleogenomics or genomics, depending. I don't, it's a Greek word, so they have their ways of to pronounce their words. So, right, allowing scientists to extract, decipher DNA from fossils and other ancient samples to study human evolution. That's one area of study. Then you have molecular anthropology. This field uses genetic data from living human beings. These are the populations, right? Where they can reconstruct the evolutionary history of a specific human species. With these, all of these studies areas are legitimate studies with technologically advanced tools that will allow them to get accurate details and time markers of the fossils when they came about. What they are missing is where our doctrine comes in, and the father, right, our father, Dr. Malachi York, makes it clear of the jumps or the interventions of genetics within the gene pools that scientists say, well, we don't know how this DNA disappeared, or we don't know how this particular DNA upgraded a group of people for where they become Homo erectus to Homo sapien, or from upright man to intelligent man, right? Or how the specific primate genes are almost similar to the human genome. Your baboon, your gibbon, your orangutan, and these other primates. Then animal genetics as well. So they see that there's an intervention of genes that are foreign to the haplogroup that from the root C, which is the SANS. They just can't explain it. So what we will do is try to explain it and, excuse me, tie it in to where you are able to understand where the missing link, because they always say, when you look at scientists, they'll say, oh, there's a missing link. There's a missing link to human evolution. You'll see this chart where you have a uh, uh, chimpanzee halfway upright, walking on all closely all fours, hunchback. Then you have one with a tool where they pick up the tools. Then you have a so-called caveman upright. Then you have this Homo sapien, right? But there's a missing link in there, and they're trying to figure out how or why that particular missing link, because they were not there when they were being created. That's why scientists don't know. You have to receive that affirmation from a being who was responsible for the genetic breeding. And that's why our deities in flesh, our God in flesh, has come to make it clear for us, because their records now being uncovered, they don't have the clarity to explain to you where they come from, let alone explain to you where you come from. So this is our job. So the molecular anthropology is where, please share the room, please. So people are not aware that we're on, and please subscribe. So we're going to get into some real good stuff here. The molecular anthropology is the field uses genetic data. Genealogical studies, which is include, oh, I went through this, right? Okay, so now let's deal with genealogical studies and how they study the genes. This is just brief for you to understand how they acknowledge scientists and how you can confirm that the Father, what he teaches is all wise, right, and exact, 100%, irrefutable, indisputable, intransgressible, right? Information that's coming from beyond that stars, the heavens, down to us, to his children. And I'll give you some authorities from their school, because a lot of people say, well, I don't find it in this reference. They need the confirmation from the Caucasoid, which is fine. That's who taught you. It's not a problem. He confirms our doctrine as well, so we won't have any issues. Now, there's a multi-regional evolution. Okay, in genetic studies beginning in the 1980s, provide explicit support for a recent origin of modern humans in Africa 200,000 years ago. This is one of the consensus across the board. They acknowledge that there's an Africa out of Africa migration that took place. They just don't know exactly at what point and what time frame, because they're getting it from paleogenomics and molecular anthropology. That's all they can compile. They're not getting it from the source, which we are getting it from the source. And you can find that study, complete book, in the Mitochondrial DNA Human Evolution, in an article called called Nature in 1987. They started printing this information, right? Also, the evolution and origins of modern man still is a current book. 19 is current. It's the latest, one of the latest, even though it's a few years old. It's the latest insofar as, not the latest. Let me say this: it's the earliest, but still the most factually verifiable. Meaning, no other books or authors have come back to debunk this particular writing by Distel Tr Distel. That's the author. Now, in this article, the recent African genesis of humans, genetic studies reveal that African women of 200,000 years ago was our common ancestor. That's in an article by this same person, the recent African genesis of humans. And the main points of this article, I'm not going to go through the whole thing, but the main points of this article, what they said is that paleontologists had argued that evolution, or the evolutionary split, happened between humans and great apes 2.5 million years ago. Now, we'll get into that because they're speaking about a specific gene pool. Now, we understand that there were beings who did intervene, right, from the Sirius star system as well as the Pleiadian star system, right? We'll get into that, the biblical texts or the records that speak about these beings coming down, the sons of God coming down to the planet, right? Also, there's a breeding process where these group of Canaanites were bred for and a disease was placed in their blood called the disease of leprosy, or leproma, Hansen's disease. That was a specific breeding. Then you had a breeding of the Adamite, which is the Genesis breeding that happened 450,000 years ago. That's a specific breeding. So there's several breedings of the Caucasoid and his line. The latest breeding was the Adamites. That's the Genesis story. These beings, Adam or Edom, which is the red ruddy race. This is the wicked race spoken of in Genesis chapter 6, verse 6 and 5, where their Lord repented for breeding this particular being, right? That was 6,000 years ago. But obviously, they're trying to find out how Africans, how we were here prior to that, and how our genetics mixed in with a certain stocks of the Caucasoid, which you'll find out that our blood was used in the breeding process as well from these Sands and the Sands people of Botswana. But this article speaks about this revelation that we all stem from an African woman 200,000 years ago. Now, geneticists argue against it, claims that the similarities were too similar to be more than a few million years ago. So they basically were trying to understand, how is it that one group, one stock looks like it came older and then another stock looks lesser? That's because of the extraterrestrial intervention and the millions of years by which these beings were coming back and forth on the planet. They don't have those records of what you'll call angels, Elohim, whatever name you use, depending on the language. Muslims, Christians, and Jews get caught up with this. Oh, they're Mala'ikat, or they're Elohim, or they're angels, little fat white babies with wings, right? The whole wing thing comes from the seraphim and the cherubim, which is coming from Mesopotamia. Father Abraham coming from Ur of Chaldees. So they got it from the same concept. The problem is they're not sure at what timeline these extraterrestrials, angels, beings from the heavens, the stars, intervened and placed their genetics to upgrade this man. So this is a lot of times when you read these articles where they question the timeline, it's because they're not aware of that intervention. So now, today, this was old. The main point of this article, where they were arguing now with this new genome. Let me turn this fan off, please. Now with this new, please share the room. Keep sharing, right? Because this is going to go really viral, and then this podcast is going to blow up because this doctrine is profound, and the world needs to hear what the Father says to his children, right? So when you so this new paleontologists today say now modern humans evolved from the archaic forebears around the world over the past million years, right? Geneticists are using maternal lines of descent to a woman who lived 200,000 years ago in Africa. Modern humans arose in one place and spread elsewhere. That's on page 68 of that particular article. The article is the recent African genesis of humans. Genetic study, or in that on page 68, you'll see throughout this article, you'll see that. Um, I'm just making reference to one article here. Now, the question comes up, why does genetic information of living subjects, you all, people who are alive, like the modern day population, like one of the brothers, Second Cosmos, right, had his DNA done. Many people use these DNA companies that track your DNA and give you your mitochondrial DNA, your matrilineal lineage. So, because living genes must have ancestors, whereas dead fossils may not have descendants. So they need your genetics, right, to confirm the descendants of the fossils. And then they can make the comparison. That's the only way you can create a phylogenetic tree. You have to have living descendants. Otherwise, if you don't have living descendants, you can't determine where the genes are today. Therefore, you can't mark them or give them a time record. So, therefore, molecular biologists, which, right, know that genes they examine must have been passed down through lineages that survive to the present. Paleontologists cannot be sure that the fossils they are examining does not lead down to an evolutionary blind alley. That's why they need you. So all of these studies are needed to create a complete chart. I hope you follow me. Let me know you're following and understanding what I'm saying. Just type in yes in the chat, and I hope you're sharing this. Right? Okay. So what are we getting at? We're getting at the two approaches when it comes to this study. You have the molecular approach, and then you have the paleographical approach. The paleographical approach is the study is considered, right? They consider this spotty. Why? Because a handful of surviving bones may not represent the majority of organisms. So basically, right, when you have a group of limited fossils, it's difficult to say, well, this fossil represents this particular population. So it's spotty in that way. The molecular approach is free from limitation that paleontologists have. Why? Paleontologists have the issue of just bones, fossils, right? But molecular approach is that they can compare the fossils and bones to the living DNA and have a more complete story. This is how they can track you through your test, because the molecular approach is that we have your molecular genes, your structure, your biological makeup, your entire genetic sequence from all of your bloodlines, right? And with that, we can determine everything about you. Paleontologists can't do that. So the molecular approach is better. Also, it is not the molecular approach is completely objective, only concerning itself with complete sets of genomes, holding all the biological information of an individual. All variants within the genome that appear within a population can be studied. 4, genomes are objective source of data that has not been defined by any particular evolutionary model. So every genome that we receive from this person, that person, is totally free of any labels from outside sources. And so when you have objective data, that means that it's not influenced by what other people say. Okay. Okay, so great. All right, so let's move forward. Um, what is DNA of the mitochondria? Why is it studied? Okay, so now Now we have many breakdowns of what mitochondrial DNA is. Um, the reason that scientists use mitochondria, and Baba explains this in the Black Book and many books, and we'll get into um, what the mitochondria is. But I'm giving to you the science insofar as why they use it to study the lineage, right? The structure of the mitochondrial DNA comes from or composed of two small stranded rings. So inside the cell of the base, the body of the cell, you have the nucleus, and then you have the body of the cell. You have DNA in the nucleus of the cell, but the mitochondria in the DNA in the mitochondria of the cell, it's a self-regulating machine. So basically it converts food to energy. So it's a constant. It's like a source. It's like an engine that is totally indifferent between the rest of the cell, meaning it doesn't need the rest of the cells to operate. Right? It's just it lends itself its own operation, and so it's like a free thinking machine. And so the reason why they use it is because it's the sequencing that you have in these rings. It accumulates mutations, these particular mutations within the genes, and the mutations are recorded. And because the mutations happen faster than the cellular DNA, it can be recorded easier because there's not a long chain of which the mutations. In other words, the genome stretches 24,000 genomes, right? But what about the family line? How would you track mutations if they're so distant? It would take longer. So the mutations happen more readily in the mtDNA, and therefore can be recorded quicker without a long sequence. And that's why you're able to get your response back on your mitochondrial DNA from the bloodline, from your mother, right? So it says the mutations are rapid and steadily. These mutations do not alter. That's another thing. The mutations don't alter the mitochondria. So when you read the mitochondrial DNA, they extract the mutations. It doesn't alter the function of the mitochondria. Therefore, it's a record of the past, a complete record of the past. This is already confirmed by, this was already, they're already confirming what Bob has been teaching since 1967, right? Natural selection does not eliminate them. Another thing that mitochondrial DNA does, it behaves like a fast ticking clock. So if you want to keep track of something, you'd rather have a fast clock. It's like watching a video, right? And you put it on a timer, you speed it up. You can watch a movie that would take 2 hours. You can watch it in 30 minutes or an hour. Well, that's what happens with the mitochondria. It's like a fast ticking clock. So you can see how the mutations and how long they took to get to a specific genome that you have presently in your own genes. And with that, you can record how far back this gene goes. And because it's unadulterated, it doesn't change the mitochondria. It's just the best genes or DNA to use to track your lineage. Number 3, geneticists can only concentrate on the genes that have mutated the fastest, right? Therefore, mtDNA is an ideal DNA because it gets left unchanged in time and effectively neutral in comparison. Again, this is why they use mtDNA. Another thing, nucleic or nuclear DNA encodes 100,000 genes. That's your nuclear DNA. Now, you have, how many genes do you have? Total of, okay, base pairs. You have a total of 21,000 genes, right? Well, with mitochondrial DNA, the genes that are in the nucleus, not mitochondria, nuclear DNA encodes 100,000 genes, most of the information needed to make a human being. True. Mitochondrial DNA encodes only 37 genes, right? So it's easier to map with less genes. easier mapping. You don't have to map nuclear DNA. Nuclear DNA is like 100,000 as we read, and mitochondrial DNA, because it has 13 protein coding genes, 22 tRNA genes, and 2 RNA genes. So it's easier. Now, the properties of mtDNA. This is the mitochondria. This is for the feminine. You need this information so you can understand how the process of how the origin of human beings and you as a supreme being, the result of the genetics that was passed down from your ancestors that put your genes or seed in the waters in Antarctica and in the Indian Ocean. We'll go into that, how you came out of water. This is different from the Adamites. Those that are listening, those that are listening that are new, we're not addressing the Adamites' creation, the Caucasoid creation, Adam's creation. We're going to address it, but this is not the topic that in base, right? This is addressing the creation of an older race that scientists now acknowledge existed on the planet for millions of years prior to this 6,000-year-old creation of the Genesis story that's outdated now. So we're going to try to create the links there. So the properties of mtDNA, right? It's present. It's a high copy number in human cells. The average somatic cell has just 2 copies of any given nuclear gene and DNA segment, but hundreds of thousands of copies of mtDNA, 123, right? Making it easier to obtain for analysis. So that's the property of the mtDNA. Another property of it is mitochondrial DNA. It's uniparental in inheritance, and is one of the great advantages of one of the great advantages of having it. It enables researchers to trace related lineages back through time, highlighting the maternal ancestry of a population. There you have it. So these are the properties, right? Another property is lack of recombination, meaning MTA DNA does not undergo a recombining. So it literally remains uncombined from parent to parent. Parent to child. And because the molecules are the same during reproduction, they don't exchange like nuclear DNA. Nuclear DNA recoils, right? Uncoils itself within the cell. This is, if you look at DNA within the cell right now, it is an uncoiled. It looks like a bunch of tubules that look unconstructed. It's only when they exchange where the chromosomes create the thread of the double helix, and then the 2 stems at the centromere exchange, and you receive 23 from each parent. The mitochondria doesn't recombine itself, so it's easier to analyze as well. So I'm just giving you the properties of mtDNA and why geneticists use it in order for you to know your bloodline, or genetics, not you to know, well, yeah, you to know, as well as geneticists that study your lineage will know how these markers are obtainable. Another, last but not least, property is mitochondrial DNA is inherited from mother alone, unchanged except for chance mutations. Excuse me. Okay. So what are these chance mutations? Please share the room too. Share it, not the room. We're not on Clubhouse, but we're on our podcast. Share if you can to those different platforms, and allow subscribe as well, and like. Okay, so you have the mutations. Now, this is giving an example of how they determine how geneticists determine the population's descendancy. A prime example is this. Imagine a static population that contains 15 mothers, right? Every generation contains 15 mothers, producing 15 daughters. Some mothers will fail to produce a daughter, whereas some will produce two or more. The maternal lineage dies out whenever there's no daughter to carry on, only a matter of time before all but one lineage disappears. So when you look at the mothers, this is something the father has been teaching us. We'll go over it in the actual facts. I'm just pulling out these articles that confirm everything that the father has been saying, that we don't need their confirmation, but scientists are now depending on what the father is expressing in this doctrine in order to tie in the missing links. So if all of the, for instance, when you deal with the population of mothers, they know that it all comes from one mother because most mothers don't give birth to daughters. They do, but as the population increases, the probability of a mother having a daughter is equal. It's obviously 50-50, right? Maybe a little bit more because more daughters are being born now. But at a specific time, the chances of, maybe it's a 30% chance, 60%, 70, 30 to 40% chance, 60 to 70% chance that they will have daughters, 30 to 40% chance that they won't. In the percentage where they don't have daughters, that means that particular lineage dies out. So then that decreases the pool of mothers. As the pool of mothers decrease, the lineage becomes more narrow. So lining it to a direct group of women is more reasonable genetic wise than dealing with the father, because the father's the father's DNA gets basically chopped up. It does get expressed, but only through the Y chromosome. So I'm just giving you the formulas and how they approach this, so you can kind of be aware of why they use the mitochondrial DNA, and not just because it's, people use this mitochondrial DNA, but you really have to know the reasons why, the properties of it, and the mutations that are involved, the clock record, the clock record, that the mutations that it keeps a record of allows it to be sequenced and studied over a larger period of time, as opposed to the DNA of the nucleus, where the cells mutate. The mutations happen over a longer period, which creates a longer genome, and the sequence takes much more time than a fragmented area where the mutations are steady. I hope that makes sense, but I'm trying to deliver it as much in a layman's term. Now, there's a key phrase is within the genes. Now, the question would be, how or what area of the gene is studied to compare to others to determine what group you all will have come from. When you do your mtDNA test, they always say the most the area by which you most probably would be from, right? Rahobat. For those that are coming, I appreciate your input. They always say, well, you might be from this particular Nigeria. You have this percentage of Nigerian, this percentage Cameroonian, this percentage of, let's say, Ghanaian, right? What does that mean? That means that they're not tying you to a specific country. Let's be clear. When they say I'm from Cameroon or from Nigeria, they're not saying you're from that demarcated area, because remember, these places became demarcated after colonialism, during independence. So when you see Nigeria, 1960, I think it became independent. Excuse me, Nigerian friends, if I'm wrong, right? But 1960, 57 for Ghana, most of your African nations, 50s and 60s and the 70s, right? They're not saying you're from that place. They're saying that the gene pool by which they've done the genetic test, that particular percentage of your genes come from this particular area. Now, the beings or the people in that area don't necessarily have to be Nigerian. They could have migrated to those places because there's migrations that took place from Botswana, the Sands people, up the Nile, right? To the Neolithic people, into Kenya, into Tanzania, into Yemen, Ethiopia, right? Saudi Arabia, then into West Africa. So there were migrations that took place. So when they say you're from this particular area, what they're saying is the gene pool, the genes, your genome structure, your sequence, right? Your restrictive fragment length polymorphic. It's RFLP. It's a restrictive fragment length, right? Polymorphism. That's an area of your gene that they study. And if there's alterations within this restrictive area, then they know they compare it to others. And they say, okay, this comparison is here, percentage is that, this percentage is there, that percentage is there, this percentage is there. And so that's why you have the percentages, because the other populations by which they did the test from that particular area has the same genome structure, or a particular area, fragmented area of genetics that you have the same as theirs. So they'll say the percentage of what you have is Nigerian, or the percentage, 78% is this and this, not the map. So don't get it confused where you think you're Nigerian. No, it's just the migrations of the population of genes that settled in that particular area. You have the genetics. So I hope that's clear, but that's how that comes about. And so remember that phrase: restriction, fragment, length, polymorphism. Now, this refers to the differences, variations amongst people in their DNA sequences. We all have these variations. When you pull up a chart of genes and your genome structure, you have geneticists, they do the sequencing where they can find out through your base pairs where if a particular fragment is sequenced with this, then they know it's the same person. That's what they do with criminal forensics, how they can tell if a person genetic DNA, if they left a DNA at the crime scene. They will take that specific area. That specific area is the restriction fragment. They take that fragment, right? And they take the length of the fragment and how it has morphosized in the fragment, compare it to this particular DNA. If the same fragment and the polymorphic changes are equivalent, the variations are equivalent, that's the same person. It's undeniable. And they do that through populations. It's not so common with populations because they have an advanced study or advanced sequencing that they do nowadays. But this particular restriction fragment length polymorphism is something that's done within the criminal field. Now, what does it do again? Let's go over it. It compares the variations amongst people in their DNA sequences as sites recognized by restriction enzymes. Restrictions enzymes, this is DNA talk, is a protein, that acts as a biological catalyst, accelerating chemical reactions without being consumed in the process. I'm not going to get into a long explanation of that, just a basic general explanation. So you'll be aware of what they do with the mtDNA, how they take the restrictive length fragments, do the sequencing, compare it to other people in the population. If the population is dominant of that particular variation, then in your sequencing, they'll say 30% of you is from this particular area. Now, It's used. This is tool. Is used as a tool for genetic mapping diseases. Yeah, again, that's what I meant to mention. They use diseases to map diseases, not like Alzheimer's, like alcoholism, like different diseases that are in your passed down from your parents, like sickle cell. Sickle cell is a trait that they can check in your mother and your parent. And if both of those genes combine, the likelihood of that child being, if that gene is passed on from the parent, the sickle cell gene from one parent, passed from the sickle cell gene from another parent, that sickle cell gene, the combination chromosome within the child gives the child a 50% chance. So the percentage is like 50%, 25%, 25%, one parent having it, the other parent not, there's no chance. And so it's like different percentages depending on the blood type as well. So it gets real complex when you're studying the sequencing, but they do that to study diseases. That's how they know alcoholism is a disease, because typically the gene that is programmed, which the father teaches us, the programming of genetics, it takes place through light. Literally, it's a crystal that a light program, just like light programs computers, electrical pulses. In a computer, they're called bytes. Well, it's the same thing happens in your genes. It happens in the base pairs. The base pairs that you have, ATCG, right? And the combination of those determines your genetic sequence, and that combination creates proteins that grows the body, the particular organism that you will become, even your personality. So this FL RFLP is how they track the variations within the genome, right? So that was an older method, but it's still used. PCR, which is polymerase chain reaction, is another advanced method, which I'm just giving you methods of how they came up with these determinations. And they do the chart, the mapping, so you'll know that it's a real science and not something that just off the cuff that the father just put in the actual facts, right? And for Sabians, for those that know that everything he says is scientifically backed up, even where it comes to the SANS people, which we're getting to, and we're getting to that as well. So, okay, so now let's get into the basic DNA and genes, so we'll understand ourselves much more clearer. Now, when it comes to your chromosomes, we all know, well, maybe we don't all know, that there's approximately 22 chromosomes that you have. That is 20. I know it's 23. Somebody says 23. It's 22 that are responsible. They're called autosomes. There's a classification. These are your body chromosomes. You have 46 chromosomes, right? 23 from each parent. 22 of those are your biological physical characteristics. 22 from your mother, 22 of your father. The 23rd one is your sex chromosome. And it's different, right? Because the X and the Y. And this is why the father explained in the black book that the Y is 3.8 less genetic material, is because it has approximately 600 less genes within it. And this was done in the laboratory with Ninty, and how, in order to eliminate or try to perfect the genes that was altered for this new creation of the Adamite, and they used the Sans people and another tribe, the Wadziba tribe, right? A female from there and a male from the Bush tribe, the Sans people, in order to breed this new worker or this new Adamite. And that's where scientists are confused in. How did the Adamite project, how did in the blood of the Caucasian you find the Sans people's genetics? It's because they used Inky, which we'll get into, right? Inky used the genes of the Sands added their genetics, and then the Pleiadians and other beings used their genetics as well. So the combination sped up the unnatural metamorphosis or evolution the Caucasoid would have taken on his own. With the intervention of these genes from other species, it sped up their regular evolutionary cycle. And then the Duff 2012 gene was added in the various projects. There's several projects that took place. So we'll do that in part 2. Each project by which humans were bred, we'll have to address each one specifically so we can be clear, because a lot of it becomes jumbled. It's a lot of information. So we have to, our job and responsibility is to make it clear that you have the Dolphin Project, you had the Nun Project, you had the Rainbow Project, you have several, right? Several projects with different genes were used of various extraterrestrial beings. And we have to put it in a timeline and teach it in a way where it's comprehensible. So you won't have everything tied up, and understand that these breedings took place over a period of time, and each project was headed by a specific scientist and a specific group of geneticists that was responsible for a particular being for a specific purpose. Not everyone's genetic breeding purpose was the same. But when you look on a planet, everyone doesn't have the same purpose, right? And that's why you see a combination of different characteristics from different people who do different things in life, and that's all they focus on. Their whole purpose is to play sports or entertain. Their whole purpose is to be a singer or a dancer. Their whole purpose is to be a sexual gratifier, right? Or a person who's a savant, right? So you have species that wasn't supposed to interbreed that interbred, and that's because within the genes, when over a period of a few thousand years breeding, right, inside of each one of these chromosomes, getting back to the projects, right, and the Y, which we veered off from, the Y chromosome having 200 genes, the X chromosome having 800 genes, and then the other 22 with specific genes with specific codes in them that's responsible for biologically, right, organizing your tissues in a specific way that the way you come out is all in design. It's a long computer program. And geneticists now are able to go within the program and alter the specific genes, remove diseases, add certain enzymes that produce, let's say, let's say Down syndrome, right? To affect they can identify the specific chromosome what Down syndrome is. They can eliminate the sequence that creates Down syndrome and eliminate it out of the genes. Or they can design. That's why we have a book, actual fact, called Designer Genes, because our ancestors were able to do this, right? And still are. They're able to design your genes and your genetics, right? Etherically, not just physically. And it's an etheric because remember, your physical body is based upon a physical etheric matrix that's created by Panatharu, right? And from the etheric world, etheric beings are responsible for creating the matrix by which your physical body grows. So within that design and the programming of the etheric energy, it's different types of ether for general conversations. The etheric energy has a specific frequency within it that manifests itself on this side of hydrogen, which is the physical composition. Everything that your body is made of, the 99 elements, has a blueprint by which the energies, they call them chakras, the energies, the 9 seats of the African race, right? The seats by which your endocrine system operates on is on a matrix of what our ancestors designed for the African race. 8 for the Aborigines, 7 for the Hindu, the Indians, right? 6 for the Caucasoid, right? Then you go on and on to one ether, and it's a list on how it's in the physical ether, how it's described, right? But however, when you look at the genes are vibrating, or the manifestation of the etheric blueprint that your chromosomes are grown through your endocrine system. It literally, your endocrine system is your hormonal system that's responsible for growth. Your pituitary gland is over your endocrine system. Your thymus gland is your endocrine system. Your ovaries, your testes are from your endocrine system. That's tied into what? Your 9 seats. Your 9 seats are actually outlets to the etheric world. So how your chromosomes are designed is done by your ancestors, and the genetics by which your body is built upon is passed on from your ancestral line and can be activated or deactivated. But back to this Y chromosome, because of the less genetic material, right? This is how they're able to go within the genes, because of the availability of the base pairs, and the studying of the sequences and genomes gives you the actual layout of how a person will be, or how a person. That's how cloning is done, right? Cloning is done the same way. They take the genetics. They know which genes are duplicated, or what should be duplicated, or which genes you choose to duplicate. You may want to have a child with dark black eyes, or the height. They have the height gene. They have the hair texture gene. They have the blood type gene. All of those genes are available and can be reprogrammed. So that's what takes place within the physical being, the 23 chromosomes from each parent. Now, chromosomes, we said before, they're not visible. If you got any questions, please put it in the chat. Please share the room. I'm just going through a lot, so to try to get us to a point where we can understand what the Father had put in the actual facts as it relates to us as supreme beings, and how your genes are so vital, and they're just like in a miraculous infinitesimal world, microscopic world of so many amazing happenings, that if you're not aware of your uniqueness and your the specialty of the African race and the supreme beings you were created from, you're going to miss your glory. And so we're just going over chromosome information and genetic information, basic information for yourself. So chromosomes are not visible in the cell's nucleus. They're not even under a microscope. When the cell is not dividing, it is not seen. Remember, I just explained, only when the egg, the sperm fertilizes the egg, the barrier shuts off, the egg becomes a shell, they exchange DNA, the DNA combines within the egg itself, and then the moment it splits is where you see the chromosomes come together. You don't see the DNA before that, because it's scattered. It's like it looks like noodles. Perfect example. It looks like a bunch of noodles, and then when it splits, they coil, they organize to make sure that the chromosomes are tight and complete. I think it's on histones, it's called, right? At a centromere, a center point. That's the double helix. When it connects like this, that center point, right? And then it splits, and then it splits again. And then the multiplicity of the zygote, that's the name of it. Embryo, zygote, then embryo, then fetus. Right. But that's the miraculousness of the DNA. The DNA is literally thinking to control the molecular structure of this particular being, right? Through the conscious mind. The two minds that combines to create another mind. The mind of the mother, the mind of the father, fat father, other, fat her. The father fats her, makes her fat. Other, M, right? The maternal line, the sacred circle, right? Uh, of the feminine, right? The orb the primordial abyss, Nun, that she's getting ready to create, the big bang that takes place from the collision of the egg and the sperm, It's a big bang in the microscopic world. So this is a creation of another universe. And this being is being born with a conscious intelligence that combined from both parents' two minds. So this DNA had to think about this particular process. It wasn't something that was just done by chance. A lot of people think that babies come by chance. Oops, I'm pregnant. No, these are divine designs by intelligence, the mind of Panatharu that exists in the etheric realm, and they're responsible for each and every event that takes place within the physical existence every second, because they exist within the first space, the tick in the top. They're working at, because in the etheric world there is no time span. So time by which event that we can recall, they've been programmed and allowed us, whoever is born in this day and time. And this is why when you hear the Father's words and master teacher, and you're able to express yourself in with Wu Nuwap, and you have a reasoning mind, right? Because reasoning is also a being, which is another conversation we'll get into, right? And it's part two. Reasoning, right, which is Nuwapu, the reasoning faculty that you're born with, that religion eliminates or tries to eliminate. All of that is in your genetics. Reasoning is in your genes first. You don't need a Bible or Quran or Torah or a religious book to reach the egg. That's not, you don't need instructions for that. Why don't you need instructions for that? It's because it's already in you genetically. You already have the innate ability to find your way through darkness in space, racing through 4 obstacles amongst 777 million other sperm cells to get to a specific target. And the egg, contrary to what people know, is that the female's egg communicates with the sperm that she wants to fertilize with. It's not the other way around. There's a chemical makeup that communicates, right, from the egg. Scientists just did a new study, and I'll pull it up, right? When I put this on a podcast with the edits and the diagrams, you'll see, and I'll put the article in there where they acknowledge that the feminine's egg communicates with a specific sperm, right? And sends signals to other sperm. Why do you think some sperm don't make it? As others go one direction. Others get caught within the mucous membrane through the urethra as it's traveling through. Others get snatched by the immune system because they're not strong enough to withhold the attack from from the attack on the sperm from this foreign, not foreign, the sperm is foreign, from the feminine, the host, from a foreign invader. She literally has a mechanism in her that eliminates these foreign invaders and communicates with the one that she chooses to copulate with. So that's why it's important when you look at the sacred feminine about the exchanging of DNA, which is parts to, right, what we're discussing. But the conversation can go into many directions. But I'm just saying all of this to say that these chromosomes and DNA are a thinking mechanism that is responsible for constructing your body. So let's go to the next point. That's what it's called, a centromere. The construction point of chromosomes is called a centromere. That's the center part where the two sides of the helix connect. 2 arms are identified. The short arm is called the P arm, and the long arm is called the Q arm, right? That's when you see the high part is short and then the lower part is longer. Each chromosome includes a single linear molecule of DNA with genes, its own set. Genes are arranged in them in a specific order so that the gene near the tip of the chromosome 4 is one person. Oh, did I write that right? It's one person. Oh, yeah, it is. So the chromosome 4, right? What it's saying basically is that the chromosomes are so in order that everywhere you see a specific gene in every chromosome, you'll find the same chromosome in every human being, meaning that there's no alteration. So that means that that allows human beings to copulate, right? But the Rh factor, the blood types do affect um, the survival of the child. So now we understand what chromosomes are, right? We understand that 22 autosomes are your biological chromosomes, and the third, which is an allosome, which is a sex hormone, is your 23rd chromosome. Your largest chromosome actually is 19. I think it has 1,500 genes. The smallest is gene 13, actually gene 21. It has 200 genes besides the sex chromosome. Everyone has a 21 chromosome that has 200 genes, as well as the Y chromosome has 200 genes. That's the smallest chromosome. The X chromosome has 800. That's relatively mid range. But the largest is chromosome 19. It has 63,806,651 base pairs. Your base pairs are how these are they like staircases, sugar phosphate staircases that create the DNA structure. And those combinations create the specific proteins that build you. So depending on the combination, determines the variation of the human being. This is why we all look different, characteristics, personality, da 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 da. So all of that is within the chromosomes. A genome is all of the DNA distributed among the 23 chromosomes. So within the chromosomes, the DNA, a genome is the DNA in these chromosomes. That's 21,000 genomes, 24,000. Baba corrected. It's in the actual facts that we have 24,000. Some scientists say 21,000. There's a 3,000 chromosome different. It may be because they have junk DNA that they're not considering as actual DNA that is activated once your monatomic elements are in, your MAF cosette is in, and you get restored. That's a different, that activates your junk DNA. So the father obviously knows what that junk DNA is. Osminium, these platinum metals are responsible for recalibrating that DNA within your body. Scientists don't know what junk DNA is, so they may not be acknowledging it in the genome structure as just ghost DNA, but it has its value, it has its use. So we know what a chromosome is. A genome is all the DNA within the chromosome, right? Uh, two sets. A genome consists of 21,000 genes, 24,000 based on the actual facts, spread out amongst 3,073,843,747 base pairs. Human genome is inclusive of both protein coding DNA and sequences of various types of DNA that does not include proteins. So in the genome, you have protein producing and then non-encoding. Non-encoding are DNA that's just used for basic operation, right? And then encoding DNA is DNA that replicates to use for cells to replicate and do over and over and over. Some DNA does not replicate and it doesn't encode. All DNA, all body genes are not encoded to produce proteins or growth, right? Some just are operational, like nerves. Some nerve DNA is not encoding. It just operates in your nervous system or lymphatic system. It just depends. It's 24,000 genes in it. So it just depends. And it's billions of base pairs. So the combination is ridiculous as far as what genes are used, but that's what geneticists do. They study it, and there's still more work to do in that field. Okay, so in 2002, scientists reported sequences of 88% of the human genome. So that's all they have. Now, so let's move forward. Okay, any questions? Appreciate those listening. I said a lot. I'm trying to get it all out, but I don't have. If I'm moving too fast, just say slow up so you can take notes if that's what you would like to do. I appreciate everyone tuning in. I'm just checking in on the chats. Tawutek, Atum Ray Kamat, Tawutek, Aaliyah, Any questions? How's the lighting? The lighting good? Background. You see Bob over there. How's everything? Is everything good? Okay. Any questions? Any questions? Okay. No questions? Wow. Appreciate everyone tuning in. Please share it real quick. Tell people to tune in. Um, okay. So are y'all enjoying this? Just type in, put a thumbs up if you're enjoying it, because we got a lot more to cover. Um, we didn't even get into, right? We're just kind of covering the genetic aspect. So coding genes, coding, non-coding. Question. There's a question that was presented. Great, great. There's a question that presented. What studies are used to determine the origin of humans, and why is mtDNA used? I think we went over this, right? Um, we two reasons why mtDNA is used to determine lineage. A lineage-based approach, which attempts to unravel the history of mtDNA, called haplo. Now, this is where we get into um, the haplogroups. Haplo haplogroups are important. You hear people talk about haplogroups, right? But it's like, what is a haplogroup? Well, a haplogroup is, let me give you the definition, is a group of similar haplotypes that share a common ancestor with a single nucleotide polymorphism mutation. And the International Society of Genetic Glossary makes this, right, basic definition. In more biological sense, haplotypes are a group of alleles in an organism that are inherited together from a single parent. An allele is a variant of sequence of nucleotides at a particular location or on a DNA molecule. So basically, on your DNA, right? In your DNA, there's a specific area called an alleles that's transferred from a parent down. It's inherited from a single parent. And with this inheritance, based on the study of this particular fragment, right, or nucleotide, they call it nucleotide polymorphism, a mutation there. They know that you belong to this particular group if you have this polymorphism, this mutation within this specific section. They fragment or uh, analyze each segment, right? It's a single line that's inherited from the parent. If you belong to this particular uh, group based on the analysis of this allele, right? Then they place you into this haplogroup. The brother said he in haplogroup K. Some people in haplogroup M. Common haplogroups are regional based, right? So, say for instance, let me pull this up real quick. Right. So, oh, did I close this? Oh, there it is. So, say for instance, haplogroup L, right? L is the root. That's the root haplogroup. The oldest matriarch single descendancy that they found to date. It goes back to Botswana, the Sands people, and it branches off into two branches on this tree. It's called a phylogenetic tree. Everyone is a part of a particular haplogroup globally. That's why they're able to do this ancestry thing, right? You have L0 and then you have L1. L0 is this haplogroup is concentrated in mostly South African and sub-Saharan region. The L1 haplogroup are responsible for the migrations out of Africa. Those L1 group metamorphosize or create other branches that becomes labeled based on the mutations of the effects of the in the genes, the environment, the food, the climate, the different things create mutations within the genes. And as these mutations evolve over time, the nucleotide changes and it records the change as a mutation in the mitochondria. That's where they find out what mutations happen and what group you belong to based on the mutations. If you have no mutations, then you almost have a 90% haplogroup of L0, which is you're of the pure descendant or a seed of the Sans people that were placed in the seas by our ancestors in order for them to procreate on the planet, to have their children created or procreate on the planet, to be caretakers of this planet. That is where the creation of Africans come from. The genes of Panatharu placed in the dolphin and in the seeds, in the waters, right, to grow onto land, which we still do today. So you have your Ogdoad and your Ennead, which are your creative ancestral Nathar that you see on ancient Mehmet on the walls, where it speaks about these groups of beings who the original creative forces or the original deities that were grown over intervals of time, and their children are the bush or the Ta'ites, right? So these sand come from the water. Scientists don't know where they come from. They just stop from there with the sands. But then they do see that the branching of L1 goes into specific areas such as Eurasia. That's why a lot of Eurasians, they'll say the original Asians were black because they have the sands have the genetic markers of all the races. So they're responsible for the diversity of people on the planet because in their genes, the diversity was able to emerge through migrations and also through other interventions, right? Let's say this. Let's stick to the migrations because the interventions created another species, which is a newer species. But we're talking about us, the original seed. The migrations created mutations based on the movement and the food variations and the mixing with other species of the 9 you have of the Nagardinate or the Negroid race. There's 9 species. And literally within the L root, there's 9 branches of Africans. And we'll get into that in this chart. I'll put the chart up when I edit it and put it on the podcast. But you have LO, L1. Now, again, LO is the root seed for the sands. And they didn't migrate out of Africa. They have the like 90% of the haplogroup of the original mitochondria DNA that the Panatharu, our ancestors, placed in the water, and they didn't migrate out of Africa. The branch that migrated because of their movements, right, into up them now, because remember, they were in the water and they speak with the click, like the echolocation at dolphins. People right now in Africa, you still hear it even in Ghana, they'll use the back of their throat as a click. They'll use that. You hear it, right? So certain animals, not animals, dolphins and sea creatures through echolocation still use that because that click creates a vibration. The echolocation sends it back to the person. They can show the distance and show the distance of how far a dolphin is, or other whales do it as well. The echolocation communication. Well, the sands people still communicate, and because they came out of the water onto land, when the beings who you call religious people call Elohim, the gods of Genesis, when they came to this planet, what beings would they come in contact with first? Right, the sands. Right, so, okay, you can't count to 9. Okay, that's weird. So, this person is trolling. I don't know who that is. Okay, so, right, who's this person? Okay. So yeah, so what happens is right, so when these beings come to the planet, this is who they came in contact with first. They came in contact with the Sands people in South Africa. And we all know that the beings who were responsible for the breeding in Genesis, where it says, "Let us make man in our image," right? And the land, the gold of that land is good. When they came to look at the gold, they had an interest in gold because the project that the Anunnaki had, one of the Rainbow projects, to breed a being on this planet to be a worker, Enki, as a Yahweh or a Lord, was responsible for using the sand because he was amongst them, and he also needed to protect them as well. And you'll find that we'll read that in the actual facts. But my point is this: the point is that these haplogroups LO are the Sands people. And let me read something where the father explains in he explains in Pa Faek Mukwanu, right? Pa Mukminu. This is the supreme beings, right? That supreme beings, the African race. In verse 242, he says, the Sands people are the tides of Botswana that are also known as the Kalahari Bushmen who were of Zimbabwe. 249. These people have all the genetic markers that you see as traits in the human race. Sands people with traits of the African hair, Aboriginal brow, Indians dimple chin, and the Asians eyelid. So when this out of Africa theory that they acknowledge, right? They acknowledge that the Asians were actually black first, African. Look up indigenous Japanese, look up indigenous Chinese, right? Wait, few thousands of years ago, they were all of the darker skin tone, is because of the migration and them coming from the sands. When you go into, let's say, how did Ethiopia, Yemen, Saudi Arabia, the southern Yemen, the Sabaean, our land, the land of the Sabaeans, Djibouti, Egypt, they all come from the sands. This is a part of the migrations. As they were moving up, then some of them came back. So they have various time intervals where they say, well, this group came back. This is true. Some went west. And it depends. And some were used as the breeding process, which spoke about in Genesis, that the Caucasoid have no idea what took place, but though it's in their records in the Mesopotamian tablets that speaks about the creation of this Lulu Amalu, right? This Lulu Amalu, and you read in Genesis 4:2, Abel speaks about, what do you call it? Herding or keeping of the sheep. And they literally have the word sands there in the Hebrew because they were looked upon as workers by these gods, by these deities, by these beings. When they said, we have not found a man to till the ground. So contrary to what the religious world believes, when they say, oh, hey, guess what? Man was created to worship God. That's not what it says in Genesis. It doesn't say man was created to worship and praise God. The intentions of these beings or this God was to create a man. We have not found a man. So they already was aware that men existed. They already was aware that there was a beings on the planet. But they needed a man specifically to till the ground for them. Not to praise them, not to worship them, not to bow and face the Qibla or Kaaba and give Salat 5 times a day, not for the Jews to pray on the Sabbath, right? And light the menorah on their so-called Sabbath day and their holy Hanukkah and these particular so-called religious holidays, where they claim, no, that's not what it says. It says that we needed a man to till the ground. So they needed a worker. That was the intentions of breeding of this being in their image and after their likeness. And you can read it in your Bible, right? So when you get to these haplogroups, right, the haplogroup that these beings, these, let's say, Elohim found were the Sands people. There were other species that cavemen that came from unnatural nature. as well that existed, but not, we were first on the planet. That came thousands of years later, right? With certain amoebas and different evolutionary processes from different types of seeds that came to the planet. Because not only did the Sands people come out of the water, or the pygmies, you had panspermia that was done as well through the natural nature process of the seeding of planets from comets. Comets have certain enzymes, part and amino acids on these particular comets that's responsible for when they hit planets where the conditions are conducive for life and growth, then these amino acids grow simple organisms, amoebas, metazoans, and these simple organic molecules that grow and eventually evolution or evolve into simple organism, and then complex organisms, and then into living entities and microorganisms, and then into systems of biological animals, viruses, bacteria, et cetera, et cetera. That's another process. But I'm talking about the African race, the seed by which you were brought here, right? Is this particular seeding of the sands took place more than 7 million years ago. So let's go moving forward, where the Father speaks about in verse 250. They have the nappy wabar hair of the Nagard Nadu Negroids, the epicanthic folds of the eyes of the Asians. That's why when you look at the Sands people, you'll see this particular fold where they don't have a complete eyelid. And it's similar, it folds over. And you see certain Black Africans that have that same trait in different tribes, because there's 9 species of us. So you don't find all of the epicanthic fold within all of the tribes in Africa. You see some, and then you'll see the wabard hair, but these different species have different hues. But that all came from the one gene from the Sands, out of the root seed, which is the LO root seed that the geneticists acknowledge comes from a specific feminine from Botswana. And this confirms it in Pafaek Mukwanu Pamukminu. In verse 7, in physical ethers that we were talking about, the etheric part of the creation of the African race, it says in verse 7, Tasu Akakat, 9 ether on the physical means Tasu Sanafu, 9 species of 9 different characteristics of the Afafre Kau, Mungaru Negroid race. And this is in Pakatat al Akakat, which is in our language, the physical ethers, actual fact number 21. So you'll see how the Father breaks down the physical, spiritual aspect of the blueprint by which each race was built on. This is not racism. This is actual facts in understanding the matrix of the particular races and the origins of us. Now, in the human DNA, and you'll find this, I'm giving a reference for you. You'll find this in Southern African and ancient genomes, right? It's an article written. It's called Southern African Ancient Genomes Estimate Modern Humans Divergences to 350,000 to 260,000 years ago. In human mtDNA, phylogeny, the Sands people root seed are labeled haplogroup LO in Southern Africa and haplogroup L1 through 6 in Central Africa, ancestrals to everyone else. The ancestors of modern Khoisan or Khoisan expanded to South Africa before 150,000 years ago, and possibly as early as before 260,000 years ago. Another reference, these are academic genetic writings, right? The populations, quote, the populations ancestral to the Khoisan have been estimated as having the largest human population during the majority of the anatomically modern human timeline. This is in Khoisan hunter-gatherers have been the largest population throughout most of modern human demographic history. Nature Communications, Nature Publishing Group. You'll find that in the article as well. So they already acknowledge that. So we already on the same page. They're just not. We have to explain where the signs come from, and we'll get into that in intelligent design, divine design, and also how that plays a part in the Genesis story, or what beings were used to breed this new Adamite. That's in part 2 though, because that's a complex timeline that I won't have time to do in one segment. It's going to be a 3-part series for the podcast. Okay, so question. What do haplogroups represent? Okay, haplogroups represent related groups of sequences. I think we read this, that are defined by shared mutations and which tend to show regional specificity that are defined by shared mutations. Right? So, yeah. So we got into haplogroups. L, haplogroup A, B, C, and D.